in the specific, I would like to talk Biophoton as a new insight into ultra-weak photon emission as a potential communication mechanism in living systems. So, okay, in principle, biophotons are ultra-weak electromagnetic radiation that are emitted by all living organisms. The energy is in this range 1.7 to 3 electron volt. The wavelength is in the visible range, and the rate is really, really low. So we are talking about from 1 to 300 photons per second and centimeter square. And the emission starts with the light and then with the death of the organism. So in the principle, it's completely out from the bioluminescence and from other thermal luminescence that we have in usual organisms. It was measured in many kind of living organisms, going from this soy bean then to human in an experiment in Japan. And, oops, sorry. Till now, there is no really meaning of this radiation. I mean, there are two hypotheses on the mechanisms, which are the base of these biophotons. A random radiative decays of some molecules excited by metabolic events, oxidative process, like for example, or coherent electromagnetic field generated within and between the cells. So biochemical reaction involving the oxygen, however. And these two hypotheses are not mutually exclusive. So in the specific, in both theories, it's predicted that any type of perturbation on the cells, on the plants, can generate a non-specific stress and give rise an increase of this emission. And for both, and this was predicted by both the theories and experimentally observed by both. So there is no a conclusion in that sense. Historically, it's not the first time that we talk about biophotons. I mean, in the specific, they were first hypothesized by Gurewitz in the 1923. Then we go to the first measurement in 1954 by Colli and Facchini, and we have here a look of how much is weak this radiation that we want to measure. And here we arrive to the term biophoton with Fritz Albert Popp, which was investigating about it. Now, recently also a lot of groups study and try to experimentally investigate biophotons in the specific to try to discover if there are some hidden cellular communication mechanisms. In the specific, they try to isolate this some signal about this communication through the biophotons, and there are some important factors. For example, in this paper, which is quoted here, was shown as biophoton emitted by growing plants or organisms can increase by as much as 30% the cell division rate in the similar organisms. So we are talking about this the so-called mitogenic effect. So we are talking about a signal that can communicate, that can be a communication among cells, but also that can communicate to organisms, to cells, to plants, some important information. Then we succeeded to perform a machine for the detection of biophotos in plants. In the specific, here is the apparatus that we built, which has a germination chamber in black PVC. It's a cylindrical germination chamber in which we put a petri dish from 4 to 7 centimeters of diameter inside. Then we have a photocounting system, which is very sophisticated from the Hamamatsu, with a low dark current for that measurement. And then a turning filter wheel having 8 circular positions for the discrimination of the light signal, because the analysis of this kind of light emission could be an important, could show an important pattern that maybe can hide some information about the growing of the organisms, about the self-organization and other kind of things. So we have data acquisition based on Arduino and LabVIEW. And then we go, this is a picture of the apparatus that we built. Then we go to the data taking campaign that we performed. We measured the growth, the germination of 76 lentils. And here we can see the dark counts, which is also an experimental observation of the fact that the apparatus that we built is extremely has an extremely low background in dark. So we have very few electronic disturbs during the acquisition. And here is the signal of these 76 germinated lentils. The part on the left, which is going down, is the residual luminescence that you had when you put the sample from outside to inside the setup. Then we have also acquired a single beam emission, and to compare them, we rescale, and we can look that rescaling in 0.164 the beam, the single beam, because the time scale were completely different at the beginning, because are two different plans. One can find that positioning minima aligned, one can have also an alineation of the maximum. So it's a very similar going of these two measurements, but moreover, it's very similar compared to other measurements performed with seeds by other collaborations. So we are talking about signals that can have a common pattern of developing. It's not equal in other kind of organisms, because, for example, in bacteria, biophotons were measured also in bacteria, the signals are all completely different. So it seems to not have a similar structure in going. Otherwise, in this case, it seems to have. But however, we also want to highlight also the power of also the direct discrimination that we put inside, because this will be also an important improvement for the future to understand which kind of signals we are talking about, and maybe from the characteristic of each signal we can extract some information in the idea that there are some communication mechanism in these biophotonic signals. Then going on some analysis, this is population growth. This was wrong, but okay. We tried at the beginning to perform a logistic equation to describe the growth of the population of these germinating seeds. And in this kind, we can think about a number of cell growing, and considering an initial condition, we can develop a function which is described each units activating in different time in the germination of the 76 seeds. And this gives us the possibility to show that there is, however, a different moment of germinating of this group of seeds. In fact, the fit shows clearly that there are units that are being activated in different time. So could be this could either a cellular communication mechanism among them for the activation of this different, we can say, different set of seeds of the total of seeds that we put inside. Then going on, we go to the probability distribution function analysis. And here, practically the probability of a photo counting that occur, some counts that occur from t to t plus we can say tau in this kind, that is the time, which for us is 1 second, because we read the counts every 1 second, can be easily written as this function, considering eta the detector efficiency and also the mean intensity. And going on, supposing to have n atoms emitting with different phases, one can calculate the intensity of fluctuation, and also going to the photocounts distribution function, which is obtained as an average over successive starting time t of the function. Then going on from this, we can define the mean number of counts and the variance. With this definition, we have only two opposite cases where the average can be obtained in an analytical form. The first is the emission that the emission is a classical stable wave, and we have the average, which is equal to the variance. And then we have the emission, which is a Gaussian-Lorentzian chaotic light. If we have thermal sources, so for thermal emissions, with n modes of similar frequency, we have this kind of equation instead. Then going on to the data analysis and taking the background, one can observe that normalizing the dark accounts, so we have a description of the function, the experimental average, which is consistent with the dark count of data of this phototube. But when we go to observe the data analysis of the biophoton emissions, here we can find that there is a symmetrical distribution fitting like a Gaussian, and there is no possibility to discriminate really precisely among a coherent signal and a thermal mode, so a thermal signal. This is for the 76 lentils. For the pin seed, we have, in reality, it's impossible to have a good fit because we are having too short data. So taking all the data, it's very difficult. But if we go to cut part of data and take a shorter emission period, we can find a similar description. So the results of all this analysis that it's very difficult to discriminate between current and thermal states using the photo counting distribution analysis in principle. Then we go, for that reason, to the diffusion entropy analysis. Diffusion entropy analysis has a clear concept at the beginning of that is, we convert the experimental time series, so the timing in the series every second that we take, to a diffusional trajectory, and then we calculate the scaling factor. In the specific, the germinating plants are a complex system because they formed several interacting units generating a wall with a specific properties such as nonlinearity, self-similarity, and self-organization, and so on. So the seed can in fact be thought as a system that self-organizes when it begins to germinate because of the watering. Then going on from this, one can develop and build these diffusional trajectories, and with the Shannon entropy and the calculation of the probability distribution function, using some equations, we can say, we can arrive to the description of two kinds of, we can say, motions. The normal distribution, so the diffusion, so the Brownian motion, and the anomalous diffusion, which can underline some correlation, memory effects, or hidden structures. And that is fundamental because considering the fact that we have timing of memory resecting, so we have a random waiting time of tau, we can reconstruct a wake non-stationarity condition that it's this condition which we have here that give us the possibility to highlight some crucial events, memory resetting, and sign of possible correlation of biophotonic emission. And we have this value if we have a normal distribution, so nothing is happening inside. So in principle, we have two possibilities at the end of this. We are or in a weak non-stationarity condition, and there could be some crucial events in this signal that we are observing, or we are in a normal distribution, so we are stationary and the signal is completely causal and not organized. Then we performed the analysis and considering starting from the dark without using of stripes, because they are also the using of stripes, we get the no correlation for the background, but we get some crucial events in some way in this pattern that we have here, as you can see, distributed among 2.29 and 2.44. This means that there is something which is happening inside, some crucial events in this signal. Then we go using the stripes, because the stripes helps us to visualize better where are crucial events. And that gives us also one more information. So we have value about 2.6 from 3.0 in this region, and then, 2.6 to 3.0, yes, in this region. Then we go to have a stable value close to 3.0 in this region. So with the stripes, we can also identify where the crucial events are happening, so in the first part, and when the crucial events are not happening, so in the last part, when the plants are going to die because there is no light inside. So the conclusion are these. The counts distribution analysis is not able to assess a clear evidence of coherence and non-classical behavior of biophoton emission. But the DIA analysis reveals the presence of crucial events that could be fundamental to understand if there are some communication mechanisms inside this signal, which in fact can have these studies. For example, the analysis of electroencephalogram in humans shows that the human brain in good health generates crucial events for mu equal to 2, instead for mu equal to 3 corresponds to a pathological state. So maybe this information could be the key to arrive to understand important physiological situation from plants to cells, and can estimate how is going the life of this cell, the life of these plants, for example. So the study is so important because can help to evaluate also the healthy condition of living organisms. These are some papers which we published about these studies that we performed. And moreover, I would like to mention a new study that we are performing at Fondazione Pisana per la Scienza, in which we measured the biophotons emitted by cell samples of astrocytes cultures and glioblastoma cultures. Successfully observed, you can see here the cells and the apparatus inside the incubator. And then we go for the measurement. We can say we have not the data because the analysis is going on, so no any conclusion or result. But there is a clear visible different rate when we have inside the cells and the dark counts. So this will give us the possibility to say that we probably can extract from here, using the diffusion entropy analysis, the same information that we got from the plants. And that could be a great result. But this we will talk maybe next year about this. And that's all from me. Thank you for your listening. Okay.
Thank you so much, Luca. If there are any questions on this very interesting topic?
I have a question. Are you able to perturb the system somehow? Is it possible that they respond to the biophotons if you emit them in the same frequency and somehow with the same structures, if you think there is a structure in the emission?
I mean, in my opinion, there is. It's sure that there is, because the study of Gurswitz at the beginning, in which he put some quartz window among two onion roots, and they communicate in some way. When you put quartz inside, the only way to communicate is radiation emission, electromagnetic and weak radiation emissions in the visible. So in my opinion, for the data that we have, starting from there, after there is some communication mechanism. You can, and the other things that it's really interesting is that it's true, when you perturb the systems, the emission, the rate of the emission is bigger. So it means that it's something is happening inside and can be communicated. You can perturb it.
You are perturbing them chemically or with the same photon emission?
At the beginning, chemically. And this can be done, maybe you can change the pH of the water, you can change other conditions and see what happens. It's expected changement, so something happens, but you can study it. Using some emissions of frequencies, yes, it's possible, because, for example, the experiment was quoted. They used some biophotons of the same frequency and observed this mitogenic increasing, and that could be really possible, because once, if this is a communication mechanism among cells, once you know how it works, you can communicate as one of them, in principle. So surely you can, in my opinion.
Luca, I didn't understand. You mentioned just at the end the potential effects of your investigation on electroencephalographic signal. Could you be more specific about that?
This was a quotation that I performed only to see how much could be important to use the diffusion entropy analysis and on this kind of signals, because that studies were performed using these encephalograms and using the diffusion entropy analysis, extracting these crucial events and look what happens to the patients. So it's completely separated, but what we are doing now. I can provide you the papers probably in which they talk about it, because it's not my field. So I could be imprecise and I don't want. So.
Thanks.
So you talked about how there's different biophoton emission spectrums for different types of organisms. And I'm thinking that something like a glioblastoma would be more like an undifferentiated cell. I don't know a lot about it. So would that be more similar to a signal that comes from some sort of simple organism like a bacteria, if it were to create an emission spectrum?
But
if there's like a characteristic.
In principle, we don't know. There are two. You say no.
No, bacteria have a very specific shape.
Yeah.
Maybe you know every. And in the case of the cell study, we have a different.
It's unique.
It's not so. It's not the signature of the kind of cells you are using.
You need to do an analysis to see the difference between glioblastoma and astrocyte.
Yeah.
Look just the signal, don't see my eyes.
Right.
It's not a phenomenological difference.
In bacteria, looking with the eyes, you see the difference. Because each bacteria has a kind of specific emission, which is kind of fingerprint.
But this one doesn't appear to have a fingerprint.
No name for this scale.
Not.
No.
Just look.
This
Okay,
scale
okay,
was chosen only to see that the rate is more, but analyzing in the future we can group it differently, and but we don't know. We have to go on with the analysis.
It might be that, I guess, running different kind of glioblastoma cells coming from, for example, differentiation, they might have a signature.
For sure, but it's not so evident as in bacteria. He can show the slide, but I don't know if you know.
No, maybe I can. This one, no? Right.
No, but
No, I'm not the slide here, so but I can show that after.
Very, very last question. When you talk about the residual linears at the beginning of your measuring, that comes from the fact that you open the box, or it's the
you talk about this. This
yes.
one. Yes.
Here, residual
No, it's when you are inserting, you know. You insert inside the plants, and as you can see by the scale, the lentils germinates with a few hours. So in the beginning, when you put the petri dish inside, we have some residual luminescence.
That comes from where? From
Yes.
the fact that you open the box?
No, from the fact that you prepare this outside, so you
okay.
open to put inside, but also from the sample
From the same
instead.
sample is
Yes.
coming, because the idea is that they are absorbing some light and releasing it?
It's terrible.
The sample stay in the lab.
Okay,
It's normal.
okay. So they kind of overlook it. Okay, so I guess you're done. Thank you so much.